Good morning, everybody. I am Eleonora Cappelletti, and I work in the group of Elena Giulotto at the University of Pavia. The title of this talk is Epigenetic Characterization of Horse Centromere Domains in Different Tissues and Individuals from the FAANG Equine Consortium. In this work, we use the horse as model system to investigate centromere biology. But why the horse? I want to remind you that centromeres are essential chromosomal loci that are epigenetically specified by the presence of SEMPA, that is an histone H3 variant that specifically marks functional centromeres. Centromeres in mammals are typically associated with the satellite DNA, that is tandem repeated DNA, and this is a barrier to dissect DNA-protein interactions. This situation is not the one of the horse chromosome 11. In fact, my group demonstrated that the horse chromosome 11 has a centromere that is completely devoid of satellite repeats. This was the first natural satellite-less centromere fixed in a mammalian species to be discovered. By immunoprecipitation with an anti-SEMPA antibody, we identified the SEMPA binding domains at horse chromosome 11, and we demonstrated that epi alleles for SEMPA binding are present. By analyzing SEMPA binding in the horse population, we demonstrated that, in fact, the position of SEMPA binding is not fixed, but slides in about 500 kilobase window, and this phenomenon is called centromere sliding. We demonstrated that these epi alleles are inherited as Mendelian traits and that sliding can occur in one generation. The first goal in the equine community of the FAANG project was to answer the question, do centromeres slide during development? To answer this question, we performed ChIP-seq mapping of EK11 centromere in tissues from different embryonic origin in 2 FAANG mares and 2 FAANG stallions, from which a tissue biobank was created. In all the 4 individuals, here there are the ChIP-seq profiles with the anti-SEMPA antibody, the position of SEMPA binding is conserved among different tissues. So there is no evidence of centromere sliding during development. The second goal of our project was to evaluate the transcriptional profile of the EK11 satellite-less centromere. In fact, it is well known that satellite-based centromeres are transcribed, but it is still under debate whether transcription is related to the presence of satellite DNA or to the centromeric function itself. We took advantage of RNA-seq, ChIP-seq, and microRNA-seq datasets produced in the FAANG effort, and we evaluated the transcriptional profile of the centromeric domain in all the 4 individuals. Here, an example is reported from one of the 2 mares with the poly-A transcript in the region of the centromeric domain, and the epigenetic landscape of this region by ChIP-seq mapping of different chromatin markers. We demonstrated that in all the 4 horses, no poly-A mRNA were present, as well as chromatin markers for active chromatin or facultative heterochromatin, and microRNAs were absent as well. So we demonstrated that transcription is not essential for the centromeric function. I want to thank the members of my laboratory, our collaborators of the equine community of the FAANG project, our funding, the FAANG, and thank you all for your attention.